Okay, welcome everyone. So thanks for joining. I'm Adrian Veres. I'm co-founder and CSO of Dyno. I'll be your host for this session. So first of all, thank you for being here. Thank you for making the trip, braving the fog and the shutdowns and voting with your feet and being here ready to connect. This session is going to run very similarly to the mix we've done so far. So expect some talks by a variety of speakers, variety of platforms, variety of problems, and then we're going to jump into discussion as well. And I will guide us through the discussion when we get there. To make room for the discussion, I'm asking you hold your questions until the discussion period so we can actually do them all together. So with that, let's get going. I also have the pleasure of being your first speaker. And I get to have a few minutes to actually dive into some more of the results that Eric previewed on some progress we've made in muscle delivery at Dyno. So developing capsids for muscle therapies. Really the reality, when I think of muscle gene therapy, it's a place where you really see this challenge that we faced over the last decades with gene therapy, where we have so much promise in terms of critical understanding, and we have such a clear sense of what we need to do, but we're somewhat held back. And even in the face of all that progress, we've actually made it to approved therapies, but we know there's a lot more to go. And what's striking for muscle gene therapies is that really all that progress has been in spite of our delivery technology rather than being enabled by it. And at the core of that is this trade-off that we have to accept in the muscle space, which is that if you look at wild-type gene therapies, they can get close to therapeutic delivery, but that requires bringing in high doses, which then bring in various forms of risks. And then you create this trade-off of therapeutic efficiency to risk. And what we need is clear, it's capsids that break that trade-off. You can have both of no safety risks and really delivery you can count on to achieve your therapeutic goals. Dyno-bn8 is our first capsid, is our newest capsid that's targeting muscle delivery. And the focus on really what we're excited about this capsid is it removes these trade-offs because we're able to achieve delivery to a vast majority of muscle cells, do so at an unprecedentedly low dose, which puts us in the best possible posture to move forward with gene therapy development. So this is really our solution to shifting this curve all the way out to the left in order to have much better outcomes for delivery. In assessing Dyno-bn8's performance, we actually felt that comparing against the wild-type serotypes would not be a good enough bar, that we needed to aim higher with other capsids in the field. We chose to compare against the published capsid, namely MyoAAV4E. And in order to have the most accurate comparison, we chose to do a head-to-head study with 3 NHPs for both capsids at doses that were all much lower than what's currently in the clinic. And we really indexed on this point of choosing doses which are up to 25 times lower than what's in the clinic because that will directly improve safety, directly improve costs to an important degree. When we look at the comparison, so I'm going to show you data that compares Dyno-bn8 with MyoAAV, and we can really see that across muscle, heart, and liver, we see improvements in the histology. It's actually a little easier when we look at molecular measurements. This isn't just in histology. In histology, if anything, because you're getting so close to everything, you actually start to blunt the change between the capsids. But when we look at measurements in vector genome biodistribution as well as gene expression, we see improvements across the board. Noting that in this design, we actually chose to use a ubiquitous promoter to understand expression across the body. In an actual muscle gene therapy, you would probably focus in on the target tissue of interest, whether it's skeletal muscle, heart, both, and obviously avoiding liver expression by just not having it expressed there. We also know that Dyno-bn8 is very dose responsive in this range. If we go up to the 1.6 E13 dose, which in this slide I'm going to call the high dose, but it's almost a strange, it's a strange statement to make. You see an improvement in the overall delivery. You see an increase in the vector genomes, the gene expression, as well as the cells transduced. The reason this matters to me is because this means it gives me confidence that when a muscle gene therapy developer takes this capsid and go into dose ranging finding studies, you'll be able to find that sweet spot in terms of where delivery will achieve the therapeutic goal for that therapy. I've shown you sort of high-level summary of the data. Let me dive in a bit, sort of tissue by tissue, muscle by muscle. So here, let's look at diaphragm and muscles responsible for limb movement. You can see consistency of delivery. Of course, for diaphragm, like critical for breathing, a place where for natural AAVs is particularly challenging to go to. Across the board with limbs, muscles for limb movement, we see this like high quality delivery that we're very excited about. We see this at the 5e12 dose. No surprise, when we increase the dose, we increase the overall level of delivery, and that's quite exciting for us. In the heart, we see good delivery at 5e12, down to about 20%. This is a very low dose. What we're excited about here is that when we go to this higher dose of 1.6e13, we are closer to a majority of the cells, of the cardiomyocytes, and here we expect that this could go up with a further increase in dose, likely for a cardiac-targeted gene therapy developer aiming in the low E13 range is probably the sweet spot and where one would start. I also want to highlight what the potential of sort of broad, even skeletal muscle delivery gives us. These are the muscles that control the face, around the eye, around the mouth. These are important for eating, speaking. They're also important to have your facial expression so you can communicate with your loved ones. You can imagine that a patient who wants a gene therapy wants to know that the delivery will be just as effective in these muscles as the ones that control motion and breathing. And with Dyno-bn8, we're excited because we really see the potential to achieve that. We've also dug deep to understand what happens in delivery to satellite cells. These are the resident stem cells in muscle. And by delivering to these cells, we actually broaden the types of therapeutic applications that we can use, because now for a gene editing therapy, where you want to edit both the actual myofibers and the satellite stem cells, you can do both, knowing that as these cells repopulate, you will continue to broaden the impact of your therapy. Finally, we've also done our homework to understand that what we see as exciting data in NHPs is expected to translate into humans. While we're not disclosing the identity of the receptor today, we've identified it. It is highly conserved in terms of sequence as well as in terms of function, and we've confirmed that Dyno-bn8 directly interacts with the human ortholog of the receptor. We've also confirmed that Dyno-bn8 shows improved transduction of various human differentiated and undifferentiated muscle cell lines. And so, voilà, this is Dyno-bn8. It's a capsid where I haven't mentioned this, I'll have one slide. You can essentially think of it as AV9 for manufacturing purposes. Between the ease of manufacturing and the low dose, this directly lowers cost. Because starting from the low dose and compounding liver detargeting, we improve the safety. And then you saw that in terms of the ability to reach both skeletal muscles and heart, we have a path for efficacy. And then based on what we know, we have a high degree of confidence in translation to humans. So Dyno-bn8 is our most recent muscle capsid. It's essentially a better version of Dyno3HV, which we announced earlier this year at ASGCT. It's joining our collection of capsids. This is Dyno-AHQ for IV delivery to the CNS in NHPs. It's also joining Dyno86M and Dyno4Z2 for ocular delivery. And the thing that's also exciting about this conference is the data that I've shown you has actually been generated using a test article that was made by our first Frontiers manufacturing partners, Trisk Bio. So if you're wondering, like, how can I ensure I get virus that's as potent to get these striking results? Well, now you know who to talk to. And we're excited that Dyno-bn8 is available for muscle gene therapy developers. Thank you.